Good morning and welcome to everyone. Please let me to represent myself. My name is Flóra Takács and I am from Gödöllő, from the Premonstratensian St. Norbert Secondary Grammar School. I recently joined to research in the Agricultural Biotechnology Center Animal Biotechnology Institute. Here I would like to thank the work of my supervisor Ellen Góca and my biology teacher Zoltán Kerényi. As I have recently joined to the research, I do not have any exact results yet, but I would like to represent what I am planning to do. The main aim of our research is to create induced pluripotent stem cells called iPS cells from fibroblast cells of rabbit. If creating iPS cells succeeds, endangered species might be possibly saved from extinction. So this figure shows that in the past 15 years the number of endangered species has risen significantly, as we see. Now the data shows that 1 of every 4 animals is in danger. As we see this figure, and we know human intervention in nature, we have to admit that only a radical solution might be effective. Let us consider some other strategies apart iPS cells. The first strategies for preserving biodiversity have focused on saving habitats. However, the magnitude of the crisis was so dire that a more direct species-targeted action is essential. The problem with artificial insemination is that only sperm can be frozen, oocyte cannot. Another problem which still exists in connection with iPS cells is that there are not a lot of donor females. Apart from iPS cells, these are induced pluripotent cells, so they are pluripotent, which means that they can differentiate into any of the 3 germ layers, ectoderm, endoderm, or mesoderm. So apart from iPS cells, there is another way to get pluripotent cells, which is deriving them from the embryo. But the problem with that in case of rodents and primates, that method was successful, but with ungulates, for example, cells derived from the blastocysts could not keep their ESC-type status after a few passage, and the exact moment of derivation could not be determined yet. Well, in this picture, so one thing is left, iPS cells. So in this picture we can see how iPS cells are created. iPS cells are derived from somatic cells, in our case from fibroblasts. We use fibroblasts because they can be removed relatively easily from the body, and they can be stored for quite a long time. For the derivation, special transcription factors are used. As I mentioned, iPS cells are pluripotent, so they can differentiate into any of the cells. So from iPS cells we can create sperm or oocyte or embryo, but you can see the question marks as there are still problems with the creation. What makes the problem is with iPS cells is to find the relevant recipient and synchronize its uterus with the created embryos. This is not only a hard task because we simply do not have enough donor females, as I mentioned, but we have very little information about embryology. In possession of this information, anyway, we could solve the problem of embryo transfer as well. So as I mentioned, we use several transcription factors to create iPS cells from the fibroblasts. These transcription factors can only result in a partially programmed cells which are not pluripotent yet. So this is just a transient population. But adding other factors, we can get iPS cells. Anyway, iPS cells are not only good in connection with saving animals, but in connection with regenerative medicine, because as you see, iPS cells can differentiate into any of the cells. What makes the problem of what is still on research in connection with iPS cells is to choose the relevant transcription factors and the method of their delivery into the chosen cell. And we have to determine also whether this transcription factor is good enough or not. For this, we first isolate RNA from the cell, and we write cDNA. And with this real-time, we analyze the expression with real-time PCR analysis or immunostaining. Then, of course, the relevant medium have to be found, and the iPS cells have to be analyzed themselves, because we have to compare them with embryonic cells. So whether are they really resemble to embryonic cells or not. During this research, I learned the basic methods, and I will take part in finding the best derivation conditions. Thank you very much for your attention.